You're listening to The Interactome, a podcast by a group of young researchers who want to connect you to the world of science by sharing their stories and perspectives. Just in case their bosses are listening, they want to remind you that the opinions expressed here are their own. They also want to remind you not to take anything they say as medical or professional advice, as they are not doctors. Not yet, anyway. Stay tuned about that. And, without further ado, welcome to the Interactome.
Hello everyone and welcome back to another episode of the Interactome. I am one of your hosts, Sarah, and today I am joined by Joe and my friend Schuyler, who is an MSTP student over at Baylor College of Medicine in Houston, Texas.
Hello.
Yes, Schuyler is working on some very, very cool research, and I'm so excited to have him on the podcast today, but I will let him introduce himself. So take it away.
Well, hello everybody. I'm Schuyler, as I was introduced. I am a MD PhD student working between Baylor College of Medicine and Rice University in a very special partnership between the two universities because because Baylor doesn't have an engineering program, so they allow some of their students to pursue their PhDs in bioengineering at Rice. So that is where my current research is, in the lab of Dr. Zablowski in his non-invasive neuroengineering lab.
Whoa, that sounds really cool. I'm super excited to get into it.
Oh, but it is.
Can you tell us a bit about before we get into kind of the details of what the cool stuff it is that you're doing right now? What's it been like for you to kind of, as you've made your way into this field and kind of grown up as a scientist in training, and just how did you get to bioengineering?
Well, I ended up in bioengineering and an MD PhD career in the same way that many of my classmates have, which is by being completely indecisive in all possible locations. This is to say, I chose bioengineering because it is a fantastic field that combines all of the strengths of chemistry, physics, and biology together into a single amalgam where you can apply basically any sort of concepts towards whatever research you want to do. Anything ranging from computer science to hard mathematics of kinematic design. Basically, wherever you want, bioengineering is included in some capacity. I started in bioengineering in college at Tufts University up in Boston. Like I said, because I was indecisive and couldn't figure out what I wanted to do between physics and engineering and biology, and fell into research in tissue engineering in the labs of Dr. Lauren Black and Dr. David Kaplan. That was a very fun time where I got to work on developing models of skeletal muscle for potential research applications in transplantation. Not actually. It's not strictly speaking transplantation. It was just research, but tissue engineering was a super cool field, but I switched over into more of an ophthalmologic bent after graduation, where I worked at Baylor College of Medicine in the lab of Dr. Benjamin Frankfurt for a while, working on a novel method to induce increased intracranial pressure in mice, and then transferred over to the School of Medicine for my medical career for 2 years, before coming to Rice University for my graduate education.
Cool. Sounds like quite a lot to get to this point.
Yeah, I'm one of the I am one of the older people in my cohort, which is fine. A lot of people who are in MD, PhD type careers take a long time to get where they're going, but it's about the journey, not the destination.
Absolutely. As someone who's also in an MD PhD program right now, I resonate with that for sure. I would say, like, for me as well, it was definitely because I ended up in this career, at least partially because I was very indecisive about doing anything else. So yeah, super, super cool though. And yeah, I guess like I think zooming out a little bit for some of our listeners, what is an MSTP specifically? Like what, like why, like, I mean, obviously we know, but I think for those of us listening who aren't as familiar, it might be interesting to kind of hear a bit about that.
Of course. So MSTP stands for Medical Scientist Training Program, and it is a specific NIH program that funds a number of institutions in their for training people who are pursuing a combined MD and PhD degree. This is essentially to pursue medical research that is particularly translational or suited towards human diseases. And the NIH believes that this is an important part of its authority and training goals, and so it funds a bunch of these institutions. Now, this is sort of separate from other MD PhDs or DO PhD institution or programs, because not all of them are NIH funded, but this has more to do with just government funding rather than anything such as a mark of quality. There are many people who are in these other programs that are doing quite fantastic research, so no shame against, nothing to say badly against them.
Yeah, totally. But then I guess, like, I don't know, it seems like you're doing a lot. Like, do you have time for, like, fun or things outside of all the crazy science you're doing? Like,
I mean, I do my best, but you know, and everybody who's in medicine or in the sciences can attest to sometimes it's hard to make sure that you're spending time for yourself. But I am a firm believer that there is at the very least, a minimal amount of self-care that everybody needs to attend to in order to succeed. I personally have been trying to work on a couple of different art projects from my fencing group, using broken weapons to build a statue and other stuff like that. But first I need to teach myself how to weld. So this, my whole, or more accurately, get taught how to weld, because that's not something that anybody wants to undergo by themselves.
Yeah.
As you can see, the indecisiveness really hasn't stopped. It's just been channeled into different places.
That's really cool, though. Like, yeah, it's, have you been fencing for, like, a while now?
Oh, God, yeah. I started fencing when I moved to Houston back in 2005. Mm-hmm. So it's been about 20 years since I have started fencing, and mostly what I've been doing recently is working as an armorer, just to sort of get the spend some time helping pay back the fencing club that I work at by fixing a lot of weapons, and it's just a fun little thing to do with my hands once a week, and then go and hang out with other fencers. What do y'all do in your spare time? Haven't really heard about that.
Oh boy. Sarah, do you want to go first, or should I?
Yeah, I'll go first. So it varies. I feel like, it's interesting, like, I'm not part of an MD PhD program or an MSTP, but I really resonate with that sentiment of just being indecisive or like wanting to do everything. And I don't know, maybe like this just a higher, maybe that trait runs in higher frequency in like those in the medical professions, but
I think it does.
It does, yeah, maybe.
Yeah, and like every single field in medicine has got something cool going on, so it's hard to really say.
That's true.
I want to do X or Y or Z.
That's true. So I think that for me that also spills over into like my hobbies and personal aspects of my life. So I'll tell you what I'm doing right now. Right now I'm trying to learn how to fly a plane, which has been very, very fun. So I'm working towards my private pilot certificate. And that has taken up a lot of my free time these days. But lately I've also been getting a bit more into journaling as just kind of like a creative outlet, and it was a way to let my mind just wander and try to have some dedicated like thinking time. So that's been nice. Although right now I am a little more pressed for time than usual with the studying. But yes, once this phase is over, I think I'm going to lean more into some of my artistic sides. I have a paint by number sitting in the corner that I really want to finish. So
super cool.
We love those.
Oh yeah. Let's see. For me, I'm not as cool as Sarah, but I still think this is cool.
Not true.
I play a lot of Dungeons and Dragons, like a lot.
Yeah.
I'm in. Technically, I was at one point I was running 3 campaigns while in med school, and that was pathological.
As the DM, no less. That's a lot of preparation you have to put in.
Well, the great thing was I was running the same campaign for each group. But
you see, it's all about those little efficiencies, just figuring out ways where you can get that extra bit of effort to stick in somewhere else as well.
Yeah.
As med students, we must not only maximize our time management during school, but we also must manage our efficiencies in our hobbies.
Yeah, I'm actually now DMing just for one campaign, and so that one's fun. And then I am playing I'm in another campaign where I play an angry space dwarf, and then another campaign where I am a depressed tiger person, because he lost his family, and it's like Greek mythology themed, and yeah, it's pretty fun. So yeah, that's my creative outlet, if you will.
Out of personal curiosity, the crazed space dwarf, is that specifically using D&D, or are you using some other RPG type system?
We're using D&D, but we're using for those who are not familiar with Dungeons and Dragons, we're using some of the Spelljammer material, which is essentially like Dungeons and Dragons fantasy stuff in space. So yeah, it's very fun to roleplay an angry space dwarf. And yeah, little different from our topic of discussion today, but I think it's like I think it's really important, and this is kind of the goal of the Interactome, to just get to know the people behind the science and like hear about all the cool stuff they do, like fencing and building cool statues and things like that.
Well, the statue part is in its nascency. We'll get to that eventually. Like I said, I got a couple of steps to complete, but in the meantime, we can talk about all of the science that's going on in the Zablowski lab.
Heck yeah. So you were saying non-invasive neuroengineering. To me,
that is correct.
Those are 2 very big words. Like what does it, like, what, maybe we could talk about, like, and obviously you're the expert here, like, maybe we could talk a bit about like what neuroengineering is, and then like what's invasive versus non-invasive. Like, I don't know what your thoughts are.
Sure. That's a fantastic way to sort of start with that dichotomy. So neuroengineering, like many scientific words, is just 2 words glommed together. So you've got neuro, so neuroscience, anything related to the brain and its function, either measuring it, figuring out what its outputs are, understanding how it works, and then engineering, which is sort of the more technical design side of research. And so the general idea behind neuroengineering is to create novel techniques and methods and instruments for measuring the brain's function, interfacing with it, or otherwise performing cool neuroscience type research.
Wow.
So this crosses a lot of disciplines. I'm approaching it from the bioengineering side, but in the neuroengineering initiative at Rice, there are electrical engineers, there are computer engineers, there are straight neuroscientists. It runs the gamut of all different kinds of scientific interests that are focused on working with the brain.
Mm-hmm.
And so the easiest way to work with the brain is to basically directly poke it with a stick, much like the easiest way to interface with anything is poking it with a stick.
Mm-hmm.
But I don't think I'm reinventing the wheel here when I say that nobody wants their head cracked open and poked with a stick. So
yeah.
The important work that's going on in the Zablowski lab and sort of the whole operating principle is that we can, using technology, figure out ways to not only probe brain function, but also understand what the outputs are and interface with the brain using techniques that do not require some form of direct assessment in terms of electronic probing or ultimately opening up the skull and using hard instrumentation in the brain to directly measure. So we look at a number of different techniques that will, through some indirect method, figure out what is happening inside of your head.
Well, I was going to say, like, I know we're about to get into some really cool methods that your lab is researching, but I also want to touch on, maybe before we get into that, what are some non-invasive ways that we currently use in clinics and hospitals to either investigate the brain or look at the brain? Because I think... Yeah.
Very fair. So the most direct, non-invasive way of measuring brain outputs would be something called an electroencephalogram. And that is a network of electrodes, so little points which can measure voltage and electricity, that are placed all across your head, not inside the skull, just on top of your head, on top of your scalp, the skin on top of your head. And these measure some amount of signal through your skull. But as you can imagine, there is a lot of signal loss because you're trying to measure something that's about maybe half a centimeter to a centimeter. I don't know the actual thickness of the skull off the top of my head. Deep to the skin. And so there's a lot of interference. There is difficulty in measuring over really small areas, and you can only really measure what's going on the surface of the brain, which is convenient for us because that is, generally speaking, where most of the cool stuff goes on. In what is called your cerebral cortex, which is the very foldy bits on the outside of the brain. That's where a lot of your higher order processing goes. But there's still a lot of interesting areas within the brain that are located deep within the brain, which control things like movement or some aspects of emotion and memory. And these you can't really measure as well with the electroencephalogram.
Yeah.
And one way to get around this is to use imaging techniques like MRI or CAT scans. And these have different issues with the level of sensitivity, which is how small of an area can they measure and on what time scale. Because MRI, magnetic resonance imaging, can measure relatively small areas of the brain, but it takes a long time to do it, and you're also looking at derived measurements of how the brain is functioning, such as looking at changes in blood flow to certain areas of the brain, which works well and does tell you a lot of different things, but it's limited by the machinery because MRI costs millions and millions of dollars and fills up an entire room and requires extensive training in order to operate it, and is a gigantic magnet that can only be used in some individuals, because if you have metallic implants, that's going to interface with the magnet and cause lots of problems. And so there are areas of improvement in ways that we can investigate the brain. Come in focused ultrasound blood-brain barrier opening. Does anybody have any thoughts, questions, concerns before I move on?
No.
Yeah, I just think it's cool.
You've really piqued my interest. So just to repeat what you said, this new way of in this case, are you trying to manipulate something in the brain, or is it a method of like kind of reading things going on similar to MRI?
I answer in the most fun way, which is to say, why not both?
Oh my gosh, that is incredible.
Oh yes.
Wait. I've never heard of this technique before. I think this is super interesting. So
no, it's a relatively new field. The technique that is sort of the centerpiece of the work in the non-invasive neural engineering lab is focused ultrasound blood-brain barrier opening.
Okay.
So let's break that term down. So focused ultrasound is ultrasound but focused, but what I will discuss first is blood-brain barrier opening.
Okay.
So listeners may or may not know that there is a specialized vasculature within the brain. So the blood vessels in your brain are not like the blood vessels in the rest of your body, because they are formed in very tight networks to prevent anything from leaking out. Just because the brain is a highly regulated space, there is a large amount of ions in the extracellular fluid and just floating around that need to be kept at specific concentrations. And there are a lot of different cell types working in tandem doing a lot of things. And so keeping the environment tightly regulated is very important for keeping your brain functioning as it's supposed to. And so your blood vessels prevent most everything, with the exception of some small molecules and organic species that have specific receptors. These are not allowed to actually enter into the brain.
Mm-hmm.
They're kept in the blood. And this is useful because, say you got bacteria in your bloodstream, it means that they're not going to be able to get into your brain that easily. And we like that because having infection in your brain is incredibly bad. And if something that had any sort of access to your body was also able to get into your brain, people would die super easily. And so this is of evolutionary advantage to keep the brain separate from the rest of the body to some degree. It just becomes a huge problem when you want to investigate what exactly is going on in there. So people have figured out that you can use ultrasound to open up this blood-brain barrier. Particularly what is used is focused ultrasound. So many people may be familiar with ultrasound from an imaging perspective. Something that's used to look at babies while they're still in the womb. That is one type of ultrasound, and it functions very similar to radar or sonar, where pulses of energy are released and then recorded as they return to figure out the depths of various parts of tissue. But it is also possible to create a focused beam. So rather than emitting hypersonic vibrations, which is what ultrasound is, it's just something that is vibrating faster than the limit of human hearing. So you vibrate from a bunch of different places and cause a specific area to vibrate at a certain frequency and intensity. this will actually cause the blood-brain barrier to, and importantly, reversibly open.
Yeah.
So the blood-brain barrier opening only occurs under specific circumstances with under-focused ultrasound, and it is a temporary process. So the blood-brain barrier is capable of regenerating itself after the opening occurs. So there is no permanent damage that is done. There's barely temporary damage that is done. There isn't temporary damage that is done. It's a safe and effective procedure that allows you to open up this blood-brain barrier and get certain species or drugs that are in the bloodstream directly into the brain.
Very cool. Wow. That's absolutely mind-boggling to me.
It is, well...
Or brain-boggling in this case.
I was going to say, it's a very direct boggling. It's basically analogy I use is shaking a present to figure out what's inside, but in a slightly different method. Especially since if you shake your present too hard, you can hurt what's going on inside, but we figured out the exact ways to specifically shake and vibrate what's going on inside the skull so that there's no damage and we can just see exactly what's going on underneath this barrier.
And how do you actually how do you see what's going on when you use this technique? Oh, go ahead. Sorry.
The technique is frequently coupled with magnetic resonance imaging, MRI, because normally speaking, you can administer what is called contrast. So molecules that produce a specific signal on magnetic resonance imaging that stay in the bloodstream and normally don't escape into the brain space. You can perform focused ultrasound and then look for areas where this contrast is present in greater quantities than it was before you performed the focused ultrasound, and that tells you where specifically you're opening.
Wow. Very cool. And what's is the utility of focused focused ultrasound blood-brain barrier opening to basically say, okay, we're giving you this drug and we want it to be delivered to a very specific part of the brain, or is it to see what are the applications, if you will, or what's the main application that people in the field are trying to work on, if any? I'm sure there's a lot.
It's being used for so many different diseases. There's dozens, if not hundreds. I would need to double check the figure of clinical trials that are going on using focused ultrasound. There was a paper that was published in the New England Journal of Medicine, which is quite a big deal among scientific journals, that used focused ultrasound to deliver anti-Alzheimer's drugs to the brain.
Cool.
So if some of you may have heard of aducanumab, which, just besides being a made-up nonsense word, is the name of a drug that's supposed to clean up the schmutz that gets in your brain in Alzheimer's. And it is administered with focused ultrasound to be able to actually get into the brain, because the structure of the chemical structure of this drug is a very large compound which otherwise wouldn't be able to access the brain. And so it's easier to administer it through your bloodstream than it would be to drill a tiny hole in your head and then try to perform a bunch of small injections into the skull. It's
Very
just
cool.
Much safer. Yeah.
Wow. So basically, you shake the blood vessel cells enough that they make a little bit of space temporarily to let these these large antibodies in this case, I believe, through. Correct me if I'm wrong.
That is correct. Yeah, it's a monoclonal antibody medication. Yeah.
Cool. Wow. Wild stuff.
Yeah. That's really cool.
Right.
Wow.
Question, kind of just taking these techniques and looking towards the future. When do you think we might see these happening in clinical practice, if we see them in that sense? You know, what would be the process of implementing that, and where are we kind of in that process of?
Well, my
all
understanding
that?
is that a major limit in the uptake of these technologies is the physical technology. This requires a very specific kind of ultrasound device to administer this ultrasound, and you often need an MRI machine to go with it, and so between those two, it is largely confined to research institutions, unfortunately. But there are independent places where you can get an MRI scan, and so theoretically this could become much more publicly available with the advancement of availability of technology. Because you need an MRI tech to actually run the thing, you need an ultrasound tech to run the focused ultrasound machine, and you need somebody to administer the drug of interest via transfusion. But transfusion clinics are super common. MRIs are expensive, which is the issue, and then the focused ultrasound device is probably not as uncommon as it could be. Like, think about basically every medical clinic is gonna, well, many medical clinics or sort of emergency care type institutions will have some form of ultrasound. In the past 5 years, there's been a huge uptake in point of care ultrasound, which is just localized imaging using using ultrasound imaging that occurs in a lot of different settings. And so as that has become more popular, you could see the propagation of focused ultrasound as well. It's a slightly different technology, but similar principle.
Yeah. Super cool.
Makes sense.
Yeah.
No.
And
So what else are you working on?
yeah, so that's just the baseline technology for the stuff that we do in the Zablowski lab. There is a lot of different potential use cases. And so the central motivating technology in the Zablowski lab, besides focused ultrasound blood-brain barrier opening, say that 10 times fast, is acoustically targeted chemogenetics. Now, I'm rattling off a lot of acronyms here just because it's, science, so it's super easy to come up with very streamlined names, but I'm going to explain everything that I'm talking about as we go one by one. So acoustically targeted chemogenetics is chemogenetics, but acoustically targeted. Now, what is chemogenetics? Have to go back to fundamentals with a lot of these different fields. So chemogenetics is a form of genetic engineering or modification that is used in research that works to more precisely control what happens on a cellular level with these in whatever organism of interest. So chemogenetics basically has two parts. There's the chemical side and there's the genetic side. And so on the genetic side, basically what you do is you have some form of marker or surface protein and this may get a little bit into the weeds, but I'm going to try to keep it high level here. Some form of marker or surface protein or receptor, something that can be expressed by a cell that produces a downstream effect via some signal within the cell. Doesn't matter what it is. Now, we can modify these receptors to become ultra-sensitive to specific molecules. Make them even more sensitive than they are based on their regular composition in the body to stuff that is not normally present. And so the example of this is called it's called HMD4Q, which is complete nonsense lettering, but it is referring to a type of receptor within the body called a muscarinic receptor that is normally found within the nervous system. Mm-hmm. That is modified to not be as sensitive to what it normally is sensitive to, but actually sensitive to this drug called clozapine. But it's sensitive to clozapine in ultra low concentrations. And so by having a receptor that is sensitive at ultra low concentrations, you can administer tiny amounts of this drug to something that is expressing this receptor and get a regular level of response, like administer 1/10 or 100th of what the active dose would be for the normal therapeutic effect of clozapine, which is a drug that is used to control psychotic episodes. And so this by modifying this receptor to be ultra sensitive to an administrable drug, you can program the receptor to activate some downstream target, be it genetic, be it physical, whatever, and then directly choose how this target turns on and off by the administration of the drug. And so by applying this receptor to specific areas of the brain, like areas that are associated with memory or areas that are associated with feeding, you can turn on or off these specific behaviors with a chemical signal.
So in that case, going back to the non-invasive neuroengineering or the focused ultrasound blood-brain barrier opening, if I have a organism that has this special receptor, in addition to like the normal receptors, I could theoretically, like, if it's this protein is present in the brain, like one thing that we could do is, and maybe this is exactly what you're doing, give people the drug and then zap with all the focused ultrasound at very specific parts of the brain. You let in a tiny little bit of the drug through that area where you're trying to zap with ultrasound, and then it activates the brain in that specific area. Is that kind of like what you're thinking about, or is that?
Yeah, that's pretty close. But what if I told you that clozapine can cross the blood-brain barrier, so you don't even have to worry about that?
Oh wow,
that's cool. You can choose a chemical compound that is capable of crossing the blood-brain barrier, so you don't even have to worry about it. Which actually brings us to the acoustic targeting. So you can use the acoustic, the focused ultrasound to open up the blood-brain barrier to allow some form of vector, something that's carrying the genetic material for this receptor, to access a specific part of the brain. Mm-hmm. And so you can get expression of this receptor in certain parts of the brain without ever having to perform a craniotomy, which is where you put the hole in the skull. Yeah. And then you can control or modify the behavior of different circuits within the brain without ever having to open up the skull.
That's sick. That's so cool.
Right? It's so cool. And like this is just, that is the central thesis of a lot of the work that goes on in the Zablowski lab, is you can figure out, we can use these chemogenetic methods to combined with non-invasive brain access to modify how the brain functions. However, this is just one aspect of neuroengineering. So that is essentially the effector side. So modifying how the brain works. Yeah. We need some form of output. We need some way to actually read what's going on inside of the brain as well. Mm-hmm. And there are other sort of techniques that the lab has developed that allow us to do this as well. And the primary one of this is called RMA, or Released Markers of Activity. So RMA is a synthetic biomarker. So synthetic meaning that it is produced by the body on it has to be induced to be produced by the body. It's not something that is normally produced by the body. Actually, let's take a couple steps back. So one of the difficult aspects of studying brain function is there really isn't a whole lot that gets secreted from the brain that we can directly measure. There are a number of different compounds that can be measured that are secreted from different organs. You can look at creatinine levels to look at both renal and muscular function. You can look at not BMP. Oh crap, I'm totally drawing a blank on what it is. You can look at troponin levels to look at cardiac function. You can look at alkaline phosphatase level. These are all various chemical markers that are normally secreted by different organ systems that can be measured in the blood, which allows you to non-invasively take a small blood sample and figure out how these organ systems are doing. Mm-hmm. There doesn't exist such a thing for the brain because it's all kept inside of the blood-brain barrier. But there are ways to engineer such molecules. Ooh. Now, RMA is the sort of first line of this technology. So it is a released marker of activity, and it is the essential backbone of it is very simple. It is it is a fluorescent marker, typically, and this will make sense to the science nerds out there. It is called a luciferase, so it's the same protein that fireflies use to light up. It is produced by cells within the brain when we deliver it via a vector. And it is coupled to a signaling molecule that tells the body to secrete it from the cells. And then once it is secreted from the cells, it tells the body to push this protein across the blood-brain barrier in the reverse direction, pushing it from the brain to the blood. Yeah. So it is a marker that will that can be released by however many cells that you transduce, and you can tie it to different genetic markers or signals of activity and use that to say, hey, this section of the brain is being more active under these circumstances. This section of the brain is being more active under these other circumstances. Mm-hmm. And you can tell that rather than having to measure with some sort of electrical signal. You can measure that with a blood test.
Really cool. Follow-up question. My initial assumption was that this luciferase would be released only if you zapped with the focused ultrasound, but it sounds like that's not necessarily the case, and instead you're actually using the focused ultrasound to deliver the gene that encodes this luciferase to the part of the brain that you care about. Is that correct? Or
Yes and no. So
Okay.
the downside with using the focused ultrasound to administer this gene of interest is that there's often off-target effects.
Yeah.
So if you think of how big the brain is, your brain is, oh, let's be conservative, or let's be generous and say it's 5% of your body weight. It isn't. That means that there's still 95% of your body that is being exposed to whatever vector you're using to administer your area of interest, your gene of interest, or like the RMA. And so there's going to be a lot of background signal if you deliver it via focused ultrasound. And so is it possible that this would work well with when being co-administered with focused ultrasound? Yes. But the experiments that we've run so far have generally used direct injection to administer this. It is just an unfortunate quirk of the technology. However, we have worked on ways to make these vectors more specific to the brain and less likely to integrate into other parts of the body. Actually, no, not integrate. Integrate specifically means something that integration in terms of genetic engineering means something that incorporates itself into your genome. And the tools that we use don't do that.
Yeah.
They are non-integrating viral vectors is the term that is used.
So it's not like you're changing the permanent like DNA code inside the brain forever. It's like more of a temporary thing, if you will.
Yes, we basically give the cells a little package of DNA and say, hey, hold on to this for us, please. And the cells say, yep, sounds good. Because we have fun tools that tell the cells to do that.
Yeah. And they also don't hold on to it forever. It's like for a certain time period.
Yeah, generally speaking, it's a certain time period, but for the most part, the expression is pretty long-lived, so you don't have to worry about dropping. You don't have to worry too much about dropping off over time. As far as current research methodologies go, but current research methodologies are also limited by how long of follow-up you have with patients, and so I don't know about the viral vectors that we are currently using are assumed to be stable to an extent, but but that's also like this is based on experiments in certain animal species, which while they do live for quite some time, they don't live nearly as long as people, and so you can't. It's hard to say with certainty, hey, this thing is going to last forever. Yeah,
and just kind of going back to RMA real quick, just to make sure I understand it fully.
Of course.
Essentially, you tell the cell, hey, hold on to this DNA. It encodes this luciferase that will light up. And it will light up when it crosses the blood-brain barrier.
Yes.
And so, like, once it gets outside, once it goes from the brain to the blood, it'll light up. But it's only going to do that. It's only going to be made in areas where the brain is actually active, like where the neurons are firing or things like that. Is that a correct understanding?
Yes, just based on the genetic circuitry that we use to program these markers.
Very cool. Wow. Wild.
It all feels so futuristic that I just wouldn't have really thought of it myself. It's also not that far off, and I think that's what makes this all really cool.
I mean, yeah, this is the cutting edge of research that we are talking about, which is it is cutting edge. It's very interesting and novel. And there's a lot more stuff that we can do with these technologies. Just, keep an eye out on the journals.
Wow. Man. yeah, I'm really looking forward to seeing some of the cool work as that starts to or continues to come out of the lab and maybe makes its way towards the clinic a bit. I mean, I can imagine many different cases where, like, I've done some clinical training at this point in my medical career, and there have been so many cases where having something like this would be really, really powerful for helping us understand what's going on with patients and without having to really poke and prod people, at least in like a more physical way. And like it would have the potential to increase both like give us like more specific measurements, and also and be more precise in our manipulations of the brain, but also make patients more comfortable too. It seems like. Correct me if I'm wrong here.
No,
but,
I mean, that is a potential endpoint of this technology, but we will see where everything goes. Human genetic engineering is a very contentious topic.
That's a really good point.
For the most part, these tools are largely speaking investigational. But who knows what could be, how they could be used in the future.
Yeah. I'll also add, like, just kind of as like a broad overview, I feel like the more that I learn, or the more that I advance in my own medical education, the more I realize how much we know, and the more I realize how much we don't know. And I think certain organ systems are more heavily weighted to some degree on what we don't know. And I think the brain is definitely one of them.
Yeah.
I'd say the immune system's one of them. And it even goes far to say maybe the gut is one of them as well, like your gastrointestinal system, which is interesting because they also call that your second brain. And there's definitely a link between the brain and the gut. So I think it's really cool that there are labs out there studying this non-invasive way of really just looking at the brain, right? And of course the therapeutic drug deliveries, but I think just being able to explore more in this non-invasive manner is going to open up the door to hopefully understanding that system a lot more, and hopefully the other ones too, because everything's linked at the end of the day. But Schuyler, I mean, you and I, we met through our shared interest of space medicine. And I'm curious to know your thoughts on how all of this could apply to that field, or how you might want to apply this work to space medicine, which is ultimately something that we both want to partake in.
I don't really have specific... The problem that I have with this research is that there's so many cool things that you could do with it. The possibilities are limitless. It is... It doth boggle the mind, all of the different ways that you could repurpose these technologies and bundle them and change how they work. And ultimately, that's sort of the heart of engineering-based research. Is what we're trying to do is come up with new techniques and new methods to investigate different aspects of the human body and human physiology. That's sort of the bread and butter of bioengineering. Are we necessarily finding out the most specific information on a novel system? Not necessarily, but we are the ones who are designing the stuff that makes that stuff work.
Yeah.
Right.
Yeah. I think one question that I'd be super curious about, looking at this stuff in the context of space medicine, is if you're using focused ultrasound to open up the blood-brain barrier, is it easier or harder to open it up in microgravity? I'd be super curious.
That's an interesting question.
Yeah,
I hadn't thought about that.
I don't know if it's like really something that would change, but I think I'd be very curious to see how, if there is any difference, or like is it easier, like do you as a non-expert in this area, do you have to like change the frequencies a little bit to or like I don't know. I'd be very curious.
At this point, who knows? That's actually
Yeah.
that'd be more of a question for Sarah, who's actively working at NASA. I'm just a lowly PhD student.
Thanks, Schuyler. Thanks for putting that one. No, I mean, in all seriousness, that's a fantastic question, and I'm not sure if it's a question that we know, or if it's a question that we know the answer to. If I had to venture a guess, because I'm not sure, I'd have to do more reading on the topic myself, but I would assume that it would be linked potentially to the fluid shifts that would occur in microgravity, and how that would affect your cerebrospinal fluid column. Like I'd be interested to look more into lumbar puncture data, and then maybe based on that, seeing what the opening pressures are, and then I don't know. I mean, I was gonna say looking at any kind of data that relates to the blood-brain barrier, maybe like in terms of therapeutics that have been used, if any blood-brain barrier crossing therapeutics have been used in space. But I don't know, maybe Schuyler has more insight on
off
that
the top of
side of things, because I've been working on sands and fluid shifts, so yeah, that's more my thought process.
Off the top of my head, chemical methods of opening up the blood-brain barrier are, generally speaking, not that safe.
Yeah, right.
So that.
But in terms of, I meant more in terms of medications that could cross the blood-brain barrier, right? Like usually we think more of those. We think of like, oh my gosh, I'm blanking on the word.
No worries. I think one example like of a medication that could cross the blood-brain barrier might be like what we talked about earlier, clozapine, that the drug,
right? Like neuropsychiatric medications, right? But I think, to my knowledge, those haven't been used in space, or at least
you would know better than I.
I don't know.
Also,
I feel like
that's probably.
I feel like I'm just talking inaccurate stuff. I don't. I feel like I can't comment on this.
I don't know if it's inaccurate. I think it probably has to do more with that. That would be very protected health information.
Yeah, exactly.
Well,
so
maybe we'll have to talk to a space psychiatrist. I don't know if those exist yet, but I think
what's his name? There's a guy who is training in that.
Oh, that's so cool.
Yeah,
that's really cool.
Yeah, I mean, well, and that's the interesting thing about space medicine too. And this is actually this is like kind of a non sequitur, but this topic has kind of come up in my conversations over the past few months, and it's just kind of like the idea of protected health information in the field of space medicine, especially at this stage when you have a certain cohort of people going into space somewhat regularly, but it's the same sort of cohort. And I think as we expand access to space, I think this will still remain a really important topic, but it will become less of an issue, I'd say. But I think with such a small size of people going to space, protected health information is very, very important to keep protected, as well as privacy of who, and this is under that umbrella, but like the privacy of the individual who's experiencing a medical event, right? It is much easier to figure that out if your group is of, let's say, a crew of 4 than a cohort of 1,000 people, right? And we haven't even had 1,000 people in space ever, just flown. I think the number is in the middle hundreds range of the amount of people who've gone into space.
Some 700,
600
780 or something in that range.
Something like that, right? So we're
of that number. Emily Candarelli was just the 100th woman to go to space. So talk about health equity.
Wow.
Right.
Yeah.
And I think so it's a topic that's come up again and again. And for example, I was curious to read some case reports, right, of people who have gone to space. And those case reports are so protected for a good reason, that I realized that, like, actually I probably shouldn't be reading them, right? Because, like, there's also a need to know aspect, and the link to who that might be, whose case report that might be, could be somewhat easily deduced. So, yeah, non sequitur, but I do want to mention that as something to consider, at least in the space medicine aspect of things, because it'll be interesting to see how that all evolves and how we decide to either release information to the public or not, and how commercial companies will or not do that. I have no idea.
So if we wanted to do a little more non-invasive neuroengineering in space, there's going to be some potential challenges for really taking the information that we gain there and, like, applying it more broadly. Given, correct me if I'm wrong, just given that we have to respect people's health and their private health information.
Yeah. And actually, I space, like, medical ethics is something that I'm really curious about, and I have been curious about for a few years. And I just haven't been able to really learn about it yet. But I think that's also becoming a very important topic of conversation.
Totally. But yeah. Yeah. I think this has been a super cool conversation. Schuyler, I have to say, I am blown away by I couldn't even conceptualize this kind of work before talking with you today, and it's just a pun intended, mind-boggling sort of area of research. Though I like to think that my mind is less boggled by it now that you've described it for us and taught us about it.
That's the goal, let everybody know the cool stuff that's going on. And I mean, if there's anything else that goes on, will all be published on the Zablowski Lab, which is just zablowskilab.org, SZABLOWSKILAB.org.
Cool. And we'll put it in our episode description too, so any listeners can check it out.
Yeah.
But yeah, thank you so much for coming on the podcast.
My goodness, of course.
And yeah, I'm in agreement with Joe. I think this research is among the coolest that I've ever heard of personally. So I'm excited to continue to follow this field and see where it takes us.
Yeah, I mean, we'll see. There's plenty of time in the future.
Yeah. Thanks again so much for coming on.
Of course.
We're looking forward to seeing where all this stuff goes. And for our listeners, just our little shameless plug. Feel free to see us on X, formerly Twitter, at the Interactome, on Instagram at Interactome_media, and on our website at interactomedia.org.